Hello everybody. Today we'll be discussing about the mechanism of metabolic regulation. All of us know that metabolism requires a many number of steps and the involvement of umpteen number of enzymes. So the mechanism of metabolic regulation also involves the mechanism of enzyme regulation or the enzymes which are involved in the metabolism. I am Dr. Priya Johnson Akra from the Department of Life Sciences, Christu Jayanti College, Bangalore. Now, enzyme regulation. So, there are different types of regulation of enzymes which helps in the regulation of metabolism. Regulation of enzyme activity is very important because in order to coordinate the different metabolic processes, whether it be anabolism or catabolism. So, it is also important for homeostasis, that is to maintain the internal environment of the organism constant. It can be the homeostasis of glucose, it can be the homeostasis of calcium, it can be anything so that it remains normal. So, enzymes can be regulated by other molecules that either increase or reduce their activity. We know that enzymes have activity and this activity can be increased or decreased. So, molecules that increase the activity of the enzyme are called as activators, while molecules that decrease the activity of the enzyme are called inhibitors. So, very easy to remember. The molecules which activate or increase the activity are called as activators. The molecules which reduce the activity of enzymes are called as inhibitors. Now, coming into the regulation of enzymes, how is it done? So, there are three ways by which the enzymes are regulated. The first one is the rate of the enzyme synthesis and degradation. So, this is how it is done at the root level. Okay. So, if you do not want a particular enzyme to act, the synthesis of that enzyme itself is stopped or you degrade that enzyme itself. Okay. So, that is the first step. Second is you actually know, not you, your system actually acts on the activity of the enzyme. Okay. So, the enzyme activity can be increased or decreased as we just now discussed. Or it can also apply by the regulation of the kinetics of the enzyme. So, in simple words, kinetics means it can either target the maximum velocity of the enzyme or the Km or the Michaelis-Menten constant, or in short, it is regulating the way in which the substrate is bound to the enzyme, or it is targeting the affinity of the substrate to the enzyme. So, these are the three ways in which the enzyme is regulated. Number one, rate of the enzyme synthesis and degradation. Second one, activity of the enzyme, and third one, kinetics of the enzyme. Now, you can actually divide the regulation of enzyme into two. So, by control mechanism can be in two methods. One is the gene expression itself. So, this is a very slow control, and the next one is the rapid control. Rapid control is you are targeting at the enzyme activity. Okay. So, there are two. One is your gene expression and the other one is the enzyme activity. Gene expression is nothing but you are targeting at the enzyme production itself. So, synthesis itself, the enzymes are not synthesized at all. So, that is where the gene expression, and the next one is the rapid control, which is focusing on two aspects. There are two different methods. One is a covalent modification and the other one is the allosteric regulation. Now, in covalent modification, you have something like a particular molecule which comes and binds covalently to the enzyme, and you all know that covalent bond is a very strong bond. Okay. So, out of the enzyme activity there are two ways. One is the covalent modification and the next one is the allosteric regulation. In covalent modification, an enzyme is switched on or off. For example, like when you are sitting in dark, you want to switch on light. Okay, the switch itself, you switch it on, then the light comes. When you switch off the light goes off and it becomes dark again. Same way, the switch is something called as certain molecules like phosphorus, or it can be an adenylate group, or it can be uridyl group. Okay, so these molecules are coming and binding to your enzyme by covalent bond, and they are trying to switch on the enzyme or switch off the enzyme. Okay, and the next one is what is called as allosteric regulation. In allosteric regulation, what happens is that a molecule, it can be an activator or an inhibitor, this comes and binds to a site other than the active site of the enzyme and thus controls the enzyme. So, allosteric regulation is a regulation by a molecule outside which comes and binds to the enzyme at a site other than the active site, thus controlling the enzyme kinetics. Enzyme kinetics is nothing but its affinity towards a substrate or its velocity of the activity. These can be controlled. So, basically you remember there are two mechanisms. One is a slow control one, which is targeting on the gene expression, which is aiming at the synthesis of the enzyme, and the second one is what is called as the rapid control one, where you have two classes. One is covalent modification and the other one is allosteric modification. Now, coming to the regulating rate of enzyme synthesis and degradation, the first one which we discussed, the production of enzyme itself is controlled at the genetic level either by induction or repression. Okay? And you should be familiar with the names repressor. So, the repressor proteins are the proteins that bind to the DNA strand itself and prevent the RNA polymerase from being able to attach to the DNA and synthesize mRNA. This you would have learned under the synthesis of mRNA. So, there the repressor protein plays its role in order to inhibit the synthesis of that particular enzyme, and as you all know, enzyme is a protein. And inducers has got just the opposite role. What does it do? It goes and bind to the repressor itself, causing them to change shape and preventing them from binding to DNA. So, what happens? You are able to synthesize the enzyme. So, when you require the enzyme, the inducers comes into role. When you want to stop the enzyme, which one comes into role? The repressor comes into role. So, the slow control or the gene expression, two proteins have got a major role to play, the repressor protein as well as the inducer protein. So, therefore, what happens? The inducers allows the transcription and thus the gene expression to take place. Now, coming to the next one which we discussed, which is the rapid control method, covalent modification. Now, covalent modification is also a means of regulating the enzyme. Here, another molecule can modify the activity of the enzymes and many other proteins. So, here a donor molecules provides a functional moiety that modifies the property of the enzyme. So, the enzyme was in a particular way, whether it was a switch on or off, we don't know. What happens? It depends on the enzyme, depends on the covalent modification. This molecule can come and control this particular enzyme. So, most modifications are reversible. Now, what are the different types of covalent modification? Covalent modification itself, you have different types. They are phosphorylation, adenylation, uridylation, ADP ribosylation and methylation. So, you should know the different methods of covalent modification. There are 5 methods. This diagram explains to you how it is. So, on the left hand side, you can see the enzymes are inactive. Okay, they are inactive. So, if you want to activate it, any of these molecules comes and binds to the enzyme. The first one, the blue enzyme, okay, it is inactive. And what happens on phosphorylation? You can see the phosphorus on the top. The phosphorus moiety comes and binds covalently to the enzyme and it can be switched on or switched off. Second type of covalent modification, adenylation. This is the orange enzyme which is inactive. When an adenyl group, the green one, which comes and binds to the enzyme becomes active. The third one, the blue one, the enzyme is inactive beginning, and then what happens? The uridine groups comes and binds covalently to the enzyme and then it becomes active. And the fourth one is ADP ribosylation. The ADP ribose comes and binds to the enzyme. The purple one, the enzyme becomes active. And the last one, which is the methyl group, methylation. Okay, here you can see that the enzyme was inactive before. Once the methyl group comes and binds to the enzyme by covalent bond, the enzyme becomes active. So, we have learned 5 different types of covalent modification, which are the phosphorylation by the addition of a phosphorus group, adenylation by the addition of an adenyl group, uridylation by the addition of uridine, ADP ribosylation by the addition of ADP ribose, and methylation by the addition of methyl group. All these molecules binds covalently to your enzyme, making it active. Okay, so that is an example of covalent modification. Now, going into the detail, let us take one example of covalent modification, the phosphorylation and dephosphorylation. So, they are the most common but not the only means of covalent modification as we discussed, but the most common covalent modification is by phosphorylation, dephosphorylation. So many enzymes are activated or inactivated by the transfer of inorganic phosphate from ATP to an acceptor, for example, the side chain oxygen of the serine molecule in your protein. Of course, your protein is your enzyme. And the combination of protein phosphorylation by kinases. So this is something which you should remember. Kinases are enzymes which helps in the transfer of phosphorus group or helps in attaching phosphorus group to the enzyme, and dephosphorylation is done by phosphatases. So phosphatase enzyme and kinase enzymes have opposite action. Kinase enzyme adds phosphorus to the group. Phosphatase enzyme removes phosphorus from the group. Okay, so and it can afford a fine level of control over the enzyme activity. So kinase and phosphatase regulating covalent modification. So if you learn biochemistry, you can understand that there are lots of kinases, enzymes which control many regulatory pathways. Okay, so the covalent modification of enzyme, this is another very tricky thing. There are enzymes which get switched on by phosphorylation, and there are enzymes which get switched off on phosphorylation. So it's not a same theory for everybody. Okay, some enzymes get switched on phosphorylation, some enzymes get switched off on phosphorylation. So this is where the beauty lies. Okay, so in the first diagram you can see that the enzyme, the green one, is off. It is switched off. Okay, when does it become active? When a kinase acts on it. What is the role of a kinase? In order to transfer the phosphorus group. So when the phosphorus groups comes and binds to the enzyme becomes active. It gets switched on. And the reverse pathway, you see, the phosphatase enzyme, what is its role? Removal of phosphorus. So the phosphatase enzyme, when it removes the phosphorus, the enzyme becomes switched off. Okay, and in the second row you can see yet another type of an enzyme which is on when there is no phosphorus group, unlike the previous enzyme. Okay, the previous one switched on when the phosphorus was in there. The second enzyme is switched on when phosphorus is not there. Okay, so for this enzyme to become active, phosphatase has to act, removing the phosphorus. So you can see the kinase acts by adding the phosphorus to it and switching it off, and phosphatase removes the phosphorus and it becomes active. So this is a very important thing which you should remember. Covalent modification itself by the addition of phosphorus can be getting activated by the addition of phosphorus for one group of enzyme, while one group of enzyme becomes inactivated on the addition of phosphorus. Okay, now we will understand it better by the example of glycogen phosphorylase. For example, I have starved. Okay, I don't have enough energy. I didn't eat for 2 days. And then what does the body do? The body will ask the liver to break down the glycogen which it has stored, so that the glycogen is converted to glucose and will give you energy. But who will give the message to the liver? See, it is time to break down the glycogen. Okay, so what happens is, it is a hormone which gives a message. There are second messengers which acts and plays a role. Cyclic AMP are second messengers, and then the enzyme which is required to break down the glycogen is what is called as glycogen phosphorylase. Okay, now here you can see the red color glycogen phosphorylase is labeled as B, which is inactive. Whenever it is labeled as A, it is active. It is labeled B, means it is inactive. So I am in a condition where I need energy, I need glucose. So what happens? The glycogen has to be broken down. So which enzyme has to be active? Glycogen phosphorylase has to be active, or it has to be in the A position. Now when does glycogen phosphorylase A become active? Only when phosphorus is attached to it by covalent modification. So this is an enzyme which becomes active on phosphorylation by covalent modification. So when I require energy, when I require glucose, there is another enzyme which is controlling this enzyme. Okay, so this is where it is very important to understand. Glycogen phosphorylase itself is regulated by another enzyme which is phosphorylase kinase. Okay, so phosphorylase kinase is another enzyme which became active on phosphorylation. this phosphorylase kinase activates glycogen phosphorylase B to glycogen phosphorylase A. So, glycogen phosphorylase, when it is covalently bounded to phosphorus, it becomes active, it acts on glycogen, glycogen is broken down to glucose and you get energy. So, I hope you are clear with the covalent modification, taking glycogen phosphorylase as an example. Now, regulation by proteolysis, this is yet another way of regulating enzymes. This is something which happens during blood clotting. You all must be familiar with, I think almost everybody would have had a small cut or some wound in your life, okay? And you will be seeing that in seconds there is a clot formation in your cut. How does it happen? It involves so many number of factors which becomes activated and which leads to the blood clot. Even in your digestion processes, there are different enzymes which are regulated and activated only when you eat the food in order to increase the digestion, which are the digestive enzymes. So, you have the proteases, which enzymes which act on your proteins which you take. Okay, chymotrypsinogen, it is an enzyme in the duodenum, it is converted to chymotrypsin. So, regulation by proteolysis is an example where an enzyme will be formed, it is a protein. It is actually present in your body in an inactive form, and by lysis of this protein, by removal of certain amino acids, it becomes active. Okay, that is why it is called as by proteolysis, lysis of the protein, it becomes active. Okay, so two small segments of chymotrypsinogen are removed to form chymotrypsin. So, the inactive form is called as chymotrypsinogen, which becomes chymotrypsin. So, many proteins are synthesized in an inactive form and are only activated at a particular site on the cell or the body. And besides the digestive enzymes which we explained just now, chymotrypsin, trypsin, all these are protease enzymes which becomes active on proteolysis. The factors involved in the blood clotting circulate in the bloodstream as zymogen or as an inactive form. Only when necessary or only when necessity comes, this zymogen is activated to the factors which are active. Now, here you can see that it is a cascade of reaction. Okay, one enzyme activates another one, that enzyme activates the next enzyme. So, it is a cascade of reaction. You can see that enteropeptidase activates trypsinogen into trypsin. This trypsin itself activates chymotrypsinogen to chymotrypsin. The same trypsin activates prolipase to lipase. So, chymotrypsin is required to break down the protein you eat in your food. Lipase is required to break down the lipid or the fat you eat in your food. Okay, the same trypsin can activate proelastase to elastase or procarboxypeptidase to carboxypeptidase. So, the different digestive enzymes are activated when a particular enzyme like trypsinogen is converted to trypsin. So, this is the example of regulation of enzymes by the zymogen. So, the first proteins in the pathways are activated by trauma, for in case when you have a wound. The activated proteins then cleave other zymogens to active enzymes, which in turn cleave other factors. So, this is what is happening in blood clotting. Ultimately, the protein fibrinogen is cleaved to fibrin and the clot is formed. The last one is what is called as allosteric regulation. Now, allosteric regulation is the regulation of an enzyme by binding an effector molecule at a site other than the active site. We all know that enzymes have an active site, but here a molecule goes and binds to a site other than the active site. Okay, so the site to which the effector, so the molecule which is coming and binding and activating or inactivating the enzyme is called as an effector. So the effector binds is termed as the allosteric effect. Now, this is a classic example of allosteric activation and inhibition. You can see that this is the enzyme, the blue color is the enzyme. On one side, on the top side, you have the cofactor binding area, the substrate binding area and the catalytic area, that is what is called as the active site. On behind or the other side, you can see that is the site which is called as the allosteric site. Now, there itself there can be activator as well as the inhibitor which can bind. When an activator binds, you activate the enzyme, and when an inhibitor binds, you inhibit the enzyme. For example, the positive allosteric effector is what is called as the activator. It has got a separate site called as a positive allosteric site. Similarly, the negative allosteric effector is what is called as the inhibitor. It goes and binds to the negative allosteric site. So when an inhibitor binds to the enzyme, all the active sites of the protein complex of the enzyme undergo conformational changes so that the activity of the enzyme decreases. While an activator binds, it increases the function of the active sites and results in the increased binding of the substrate molecule. So here basically the activator as well as the inhibitor makes a conformational change in the active site of the enzyme so that the substrate binds to the enzyme or substrate do not bind to the enzyme, and ultimately products are not formed. So this is a very important topic, the allosteric activation, and it's a very long, you need a lot of explanation towards that, taking different examples. So today we have discussed about the different types of metabolic regulation by different enzymes and the different types of enzyme regulation. We discussed that there are basically two different types. One is at the gene expression level, where you actually synthesize or degradation of enzymes are stopped. Then we also discussed about the different types, which are they. One is the covalent modification as well as the allosteric modification. We also discussed about the zymogen activation. So covalent modification, zymogen activation and allosteric regulation. Thank you.